Have you ever just stopped and thought about how the trillions of cells in your body actually manage to work together? I mean, everything from thinking a thought to healing a cut, it all boils down to this invisible, incredibly complex conversation happening inside you right now. Today, we're going to decode that secret language of cells.
So, here's our game plan. We'll kick things off with the basics of how cells even talk to each other. Then, we'll look at the universal process they all use, kind of like a secret handshake. After that, we're diving deep into the key molecular gatekeepers that make it all happen, before we zoom back out to see why any of this really matters. All right, let's jump right in. To even begin to understand how something as complex as a human being can exist, we've got to start with the fundamentals, the messages our cells are constantly sending and receiving.
I mean, really think about that for a second. It's an absolutely mind-boggling question, right? Well, the answer lies in these tiny little chemical signals that travel all over the place to coordinate, well, everything. Let's break down the main ways they pull this off.
Okay, first up, you've got something called autocrine signaling. This is basically a cell sending a message to itself. You can think of it like leaving a sticky note on your own computer screen as a little reminder. The cell makes the signal, and that signal acts right back on its own receptors.
Then there's paracrine signaling. This is all about local communication. It's like whispering a secret to the person sitting right next to you. A cell shoots out a signal that only affects the cells in its immediate neighborhood.
And for the really long-distance messages, we have endocrine signaling. This is basically the body's postal service. A cell, maybe in your brain, releases hormones into the bloodstream, and they travel all the way to target cells far, far away, like in your kidneys. It's pretty amazing.
Now, the messages themselves, these molecules are called ligands. They come in two main flavors. First, you have the hydrophilic, or water-loving ones. They're happy to float around freely to get to their target, but they just can't get past the cell's oily outer membrane. Then you've got the hydrophobic, or water-repelling ligands. These guys need a protein chauffeur to travel through the body, but once they arrive, they can slip right through that cell membrane and deliver their message to a receptor on the inside.
So here's the really cool part. It doesn't matter if the signal is short range or long range, or what kind of ligand it is. Most of these cellular conversations follow a surprisingly universal three-step process. You can really think of it as a secret handshake that all cells know.
Let's break that handshake down. Step 1 is reception, where the target cell actually detects the signal. Step 2 is transduction. This is where the message gets relayed and amplified inside the cell, kind of like a game of telephone. And step 3 is the response, which is the final action the cell takes because of that signal.
And that first step, reception, is so incredibly specific. Every receptor protein is shaped to fit only one particular ligand. It really is just like a unique key fitting into a perfectly matched lock. That's how our body makes sure the right message gets to the exact right cell without any mix-ups.
Okay, so let's focus in on one of the most important and common of these cellular locks, a type of receptor called the G protein-coupled receptor, or GPCR for short. These things are everywhere.
The first thing you've got to know about this receptor is its wild structure. It's this huge protein that literally snakes its way in and out of the cell membrane exactly 7 times. And this signature shape is absolutely critical for its job as a cellular gatekeeper.
So what's this receptor attached to on the inside of the cell? Well, that's the G protein itself. And I love this analogy. It's made of 3 parts or subunits called alpha, beta, and gamma. You can just picture them clustered together, kind of like the 3 petals of a closed flower.
So in its inactive or off state, the G protein is just chilling. The alpha subunit is holding on to a molecule called GDP, and all 3 of those petals are clamped shut, just waiting for a signal to arrive.
But then a signal arrives. The ligand docks with the receptor, and boom, everything changes. The receptor shifts its shape, and that tells the G protein to swap out that low-energy GDP for a high-energy molecule, GTP. Think of it like swapping a dead battery for a fully charged one. This jolt of energy makes the alpha subunit break away from the other 2. The flower opens, and now that alpha subunit is free to get the message relay started inside the cell.
And once that alpha subunit is on its own, he can go interact with other proteins to pass that message along. Now, there are 3 main types you should know about. GS is stimulatory. It basically tells the cell, go. GI is inhibitory. It tells the cell, stop. And GQ kicks off a whole different pathway that uses calcium as a messenger. And this is so important because an estimated 30 to 50% of all modern medicines, from allergy pills to heart medication, work by targeting these very specific G protein pathways.
This is just a fantastic way to think about what happens in that GS, or stimulatory pathway. The signal ultimately activates a molecule called protein kinase A. It has a part that does the work and a part that keeps it in check. The signal basically removes the regulatory pin, letting the catalytic part loose to do its job, just like a fire extinguisher ready to spray.
Now, G proteins are definitely the most common gatekeepers, but they are not the only game in town. Cells have other specialized receptors on their surface for all different kinds of messages. Take enzyme-coupled receptors, for example. The Swiss Army knife is a perfect analogy. These proteins have two different tools built into one molecule. There's a receptor part on the outside to catch the signal, and then there's an enzyme part on the inside that gets switched on to pass that message along.
So this table kind of breaks down the main families. You've got some, like receptor tyrosine kinases, that actually have to pair up with a buddy when a signal arrives, then they activate each other. Others don't have their own enzyme tool, so they have to call one over to help out. And then, still others use a partner receptor to get the activation process going. They all get to a similar goal, just using slightly different kinds of teamwork.
And finally, we have the speed demons of the receptor world, ion channel receptors. These are essentially just gated pores in the cell membrane. When the right ligand shows up and binds, the gate swings open, and ions, like sodium or calcium, just flood into or out of the cell. This causes a super rapid change in the cell's electrical charge, which triggers an immediate response. It is unbelievably fast.
Okay, so we've gone way down into the nitty-gritty molecular details. Let's pull back out now and look at the big picture, why this incredibly intricate language is so important.
So when you put it all together, what you see is that this silent, complex language is nothing less than the master control system of our bodies. From those long-distance hormonal signals to the lightning-fast flashes of ion channels, these pathways dictate how our cells grow, divide, function, and even when they die. It orchestrates absolutely everything.
And that really brings us to our final thought. This isn't just some abstract biology textbook stuff. When you realize that so many diseases, things like cancer, diabetes, a lot of neurological disorders, often boil down to a single miscommunication, a broken link in one of these signaling chains, it just shows you how profoundly vital this cellular language is. Understanding it is, in a very real way, fundamental to understanding ourselves.